sorry, the video cut out, so I'll just pick up right where I left off. Sorry for cutting it off so quickly. I don't want to restart the whole thing. So we'll just start right where we were, which was talking about glycolysis. And when you're anaerobic and you're doing glycolysis, trying to get energy, you're end up making pyruvate. But the pyruvate, because there's no oxygen, it can't go into the Krebs cycle. You can't run it through the Krebs cycle because that requires oxygen. So the pyruvate just kind of builds up, it ferments, it turns into lactic acid, and that can then end up with lactic acidosis, which causes muscle cramps. And acidosis just generally isn't good for your tissue. So you don't want to have too much of that. So creatine phosphate is a way to kind of prevent or, bypass having lactic acidosis because it doesn't make lactic acid. It's already a stored form of energy. You don't have to go through the Krebs cycle. You already created the ATP or the energy, the high energy compound here, and you can make the ATP straight from it. So it helps to prevent that. Although once your stores of creatine phosphate are used up, then, well, you're stuck back with just glycolysis and pyruvate and fermenting, so you need to get your oxygen levels back up in order to prevent the lactic acid. So another example of substrate level phosphorylation is the Krebs cycle. So here is the Krebs cycle. We're going to see a lot of the Krebs cycle. And again, remember, you do not have to memorize every step of the Krebs cycle. That doesn't seem a worthwhile thing to do, to just sit there and memorize it. But we are going to see some of these compounds that are part of the Krebs cycle again and again and again. So they're going to become familiar to you, kind of whether you try to have them familiar to you or not. You're just going to start to recognize them, which is a good thing. It makes life easier if you recognize them. Like pyruvate. Okay, not technically part of the Krebs cycle. It's part of glycolysis. It's the end product of glycolysis, but you're probably starting to recognize it already, even though we're only on, not very far into the class at all. Acetyl-CoA, that's another one that you're probably recognizing, that you should recognize. Acetyl-CoA is the thing that goes into the Krebs cycle. And how it's entering into the Krebs cycle is that acetyl-CoA is reacting with the final product of the last turn of the Krebs cycle. So the end of the Krebs cycle is a molecule called oxaloacetate. It's another one you're going to see quite a bit of this semester because it's involved in more than just the Krebs cycle. But when we think of it just in terms of the Krebs cycle, acetyl-CoA plus oxaloacetate give us citrate. And that's the beginning. So I'll put like step 1. So this would be the most proximal part of the Krebs cycle. And oxaloacetate is the end, so that would be the most distal part of the Krebs cycle. And they just feed into each other. So you just go around and around and around like that. Another molecule that you're going to see a lot of just as we move forward with what's going on in the mitochondria and all of that is alpha-ketoglutarate, and then malate. So these guys are the ones that you want to not memorize, but pay closer attention to. Just feel like you might recognize them later. Now, I should use a different color than the one I already used. What I'm talking about here is just the overview of the Krebs cycle. I've kind of gone off on a little bit of a tangent from our substrate level phosphorylation, which we'll get to in a second. But I just want to really kind of explain what's happening in the Krebs cycle here, or the citric acid cycle, if you want to call it that. We're just having a bunch of chemical reactions. There are enzymes. You can't possibly see these enzymes here. They're very, very tiny, written down here. When you look at the slide, you can zoom in on them, but there's various enzymes that help to drive each of these transformations as we get each of these intermediary products. Now, as we do that, we're also taking NAD and turning it into NADH. We're taking FADH2 or I'm sorry, FADH and turning it into FADH2. We'll talk about those later. Those are going to go into the electron transport chain, which is how we're going to get a lot of energy out of that. So these things are important, right? Just in the end, we're going to end up, that's kind of the whole point of the Krebs cycle, is to get things for the electron transport chain. That's where we're going to get a great lot of energy. But we're also going to do substrate level phosphorylation here, which is going to give us just a little bit of energy, not a lot of energy, but a little bit. And it's here that we're just adding a phosphate to a molecule. And in this case, our molecule is not ADP turning into ATP. It's GDP, which has 2 phosphates on it. We add a phosphate to it, and we get GTP, which has 3 phosphates, and that's our energy molecule right there. So this is happening right at the succinyl-CoA part of the Krebs cycle, and as we turn succinyl-CoA into succinate, then we are kind of taking away a phosphate as we do that and adding it to GDP. Okay. Now, what happens with that GTP? It's like, okay, great, high energy compound, but what are we going to do with it? We're going to take that GDP and we're going to put it into the intermembrane space, so that space between the outer mitochondrial membrane and the inner mitochondrial membrane, and we're going to convert it into ATP. So there's our ATP that we get out of the Krebs cycle. Okay, now the ATP that we make in the Krebs cycle, in the form of GTP, the ADP that we are, using also when we're doing the electron transport chain, etc., to make ATP. We need to be able to transfer those across the mitochondrial membrane. So if we remember, there's the outer membrane, and then we have the inner membrane. And the Krebs cycle and the electron transport chain are all happening inside here in the matrix. So we need to have ADP in there, ready to have substrate level phosphorylation happen to it. And we need to get ATP that we create in the electron transport chain out. Now, it's not just so easy as that. ADP can't cross that membrane. Either can ATP. So we have to carry them across. And this is just like what you've seen before with like a sodium ion crossing a cell membrane. It needs a carrier protein to help it get across. So this is going to be really, really crucial. If you don't have ADP there by the electron transport chain ready to accept all those phosphates that you're trying to put onto it, you're not going to be able to make ATP. And then once you have made all that ATP, if you can't get the ATP across, well then there's no purpose to making all that ATP. So you got to get the ATP back out of there. So here we have the carrier mechanism to do that. So this will be our inner mitochondrial membrane. So this is our membrane and we want to get it across. Here's the intermembrane space, so that means the outer membrane is over here. I'll just draw it on there to make it look a little bit easier. So we have this whole transport mechanism going on over here. So let's start with moving into the matrix first. The name of this particular channel is a phosphate translocase channel. This is a symporter. Symport, you may have seen before as cotransport. It just means that 2 or more ions or molecules, I'm just going to write things, move in the same direction through it. So if one thing is going in, the other thing is also going in. So in this case, in the phosphate translocase symporter, we have this H2PO4 minus. It's moving in, and it's taking with it a hydrogen. So they're moving in together. So now we have H2PO4 and hydrogen inside the matrix, essentially. So here's the matrix. Okay. So we did that. And remember, the phosphate, that's going to be important. We're going to need that for creating ATP. Like, the phosphate is important for that. So we can start to exchange here with this adenosine nucleotide translocase antiporter. That's this one over here. That's going to be going in opposite directions, because antiport, you may have seen before, is countertransport. And that just means two molecules or things. I'll just write things. Could be just ions even, moving in opposite directions. Okay, so if one is going in, the other is going out. So remember, ATP is being generated in here, right? So we are wanting to get that out into the intermembranous space, and then again out into the cytoplasm. So there goes our ATP through this carrier. It's going into the intermembrane space, and it's going to eventually get out into the cytoplasm. But we're going to be exchanging it for the ADP. That's going in through that carrier at the same time. So the ATP is being made in the matrix. The ADP is being imported into the matrix so that we have ADP available to bind up with the phosphate. Okay, so when we add, we have phosphate available to add to ADP to create energy during the electron transport chain and move that ATP out in exchange for more ADP so we can make even more ATP. So it's this whole big sort of circle. These two carriers are dependent on each other, and it's helpful to be able to shuttle them across. You need to be able to shuttle these molecules across. Okay, so that's it for substrate level phosphorylation. Our next video will be about oxidative phosphorylation, which is where we get into the electron transport chain and all those sorts of things.